Hello students, today we are going to discuss some important aspects of heteroglycans. Carbohydrates are the important classes of organic compounds found in the biological systems in addition to proteins, lipids and nucleic acids. These are a set of naturally occurring compounds containing carbon, hydrogen and oxygen, with hydrogen and oxygen generally in the ratio of 2 to 1 atom as in water. These include the compounds like glucose, fructose, sucrose, lactose, starch, glycogen, chitin and cellulose. Most of the carbohydrates found in nature occur as polysaccharides of high molecular weight. Polysaccharides are also called glycans, and these differ from each other due to the factors like the identity of their recurring monosaccharide units, the length of their chains, the types of bonds linking the units, and the degree of their branching. On basis of their recurring monosaccharide units, these are classified as homoglycans and heteroglycans. Homoglycans contain only a single type of monomeric unit, while heteroglycans contain two or more different kinds of monomeric units. Let's throw some light on the classification of homo and heteroglycans. Homoglycans are generally classified as pentosans and hexosans. The chief example of homoglycans are starch, dextrin, glycogen and cellulose. The heteroglycans are, however, classified into four major classes, that is hemicelluloses, gums and mucilages, pectic substances and mucopolysaccharides. We shall discuss all classes of heteroglycans one by one, starting with hemicelluloses. Hemicelluloses are heterogeneous polysaccharides which are formed through biosynthetic routes different from that of the cellulose. Hemicellulose is a collective term used to represent a family of polysaccharides such as arabinoxylans, glucomanons, galactans and others that are found in plant cell wall. These have different composition and structure depending upon their source and the extraction method. It can be said that hemicellulose is a copolymer of different C5 and C6 sugars that exist in the plant cell wall. Like cellulose, hemicelluloses commonly function as supporting material in the cell wall. Most hemicelluloses have a degree of polymerization ranging from 150 to not more than 200 units. The amount of hemicellulose of the dry weight of wood is usually between 20 and 30%. The composition and structure of hemicelluloses in the softwood differ in a characteristic way from those in the hardwood, with considerable differences in content and composition between the stem, branches, roots and bark. Some important properties of hemicelluloses are their insolubility in water at low temperature, the increase in their solubility at elevated temperatures, their lower hydrolysis temperature than that of the cellulose, and higher solubility in presence of acid. There are some principal structural differences between cellulose and hemicelluloses. Hemicelluloses are mixed polymers, whereas cellulose is a pure polymer of glucose. Apart from arabinogalactan, which is heavily branched, the hemicelluloses have short side chains. Cellulose is a long, unbranched polymer. Hemicelluloses are low molecular weight polymers. However, cellulose has a very high degree of polymerization. Hemicelluloses may have large side groups substituting hydroxyl groups on the C2, C3 and C6 positions. The solubility and susceptibility to hydrolysis of hemicelluloses are greater than that of the cellulose. The second class of heteroglycans comprises of gums and mucilages. Gums are considered to be pathological products formed following injury to the plant or owing to unfavorable conditions such as drought, injury or breakdown of cell walls. Mucilages, on the other hand, are generally normal products of metabolism formed within the cells and may be produced without an injury to the plant. Gums and mucilages have similar hydrophilic constituents and on hydrolysis yield a mixture of sugars and uronic acids. These combine with water to form viscous solutions or gels. The nature of the compounds involved, however, influences the properties of different gums. Linear polysaccharides occupy more space and are more viscous than highly branched compounds of the same molecular weight. The branched compounds form gels more easily and are more stable because of the lack of extensive interaction within the chains. Gums and mucilages have certain similarities, which include both are hydrocolloids, both are translucent, amorphous substances and polymers of mixed monosaccharides. Both have uronic acids in their molecular structure. Gums and mucilages are obtained in high quantities from varieties of plants, animals, seaweeds, fungi, and other microbial sources. In general, plants are known to be their largest sources. The different available gums and mucilages can be classified into various types. For example, according to the charge, as non-ionic gums like guar gum and locust bean gum, and anionic gums like gum arabic, caraya, tragacanth, etc. According to the source, as marine origin like agar and carrageenans, plant origin which includes exudate gums like gum acacia and gum ghatti, or seed gums like guar gum and locust bean gum. Animal origin which includes chitin, chitosan, chondroitin sulfate and hyaluronic acid. And last but not least, microbial origin, which includes the gums and mucilages obtained from microbial sources like xanthan, dextran, plulan, schizophyllin, lentinan and crestin, etc. According to monomeric units in chemical structure, gums and mucilages are classified as diheteroglycans like algins, carrageenans and galactomanans. Triheteroglycans like arabinoxylans, gellan and xanthan. Tetraheteroglycans like gum arabic and xylem seed gum, and pentaheteroglycans like ghatti gum and tragacanth. Now let's talk about some important applications of gums and mucilages. Firstly, applications in food industry. Gums and mucilages have a variety of applications in the food industry. Different gums have different uses. For example, water retention and stabilization is the property of guar and locust bean gum. Stabilization for ice cream, meat products and instant puddings is the property of carrageenans, while stabilization of dairy, confectionery, beverages, baked products and sauces is the property of gum arabic, tragacanth, pectins, alginates and xanthan gum. Secondly, pharmaceutical applications. Gums and mucilages have a variety of applications in pharmacy. They are used in medicines for their demulcent properties for cough suppression. These are useful as tablet binders, disintegrants, emulsifiers, suspending agents, stabilizing agents, thickening agents, film forming agents and sustaining agents in matrix tablets. Thirdly, industrial applications. Gums find applications in different cosmetics, textiles, adhesives, lithography, paints and paper manufacturing. There are several advantages of natural gums and mucilages. For example, these are biodegradable, having no adverse impact on humans or environmental health. These are biocompatible and non-toxic. The production cost is much lower compared to that of synthetic materials. These employ an environment-friendly processing. These are locally available, especially in developing countries like India. The third important class of heteroglycans are pectic substances. Pectins are polysaccharides occurring in all plants, primarily in their cell walls. These act as intracellular cementing material that gives body to fruits and helps them keep their shape. When fruit becomes overripe, the pectin is broken down into its monosaccharide constituents. As a result, the fruit becomes soft and loses its firmness. One of the most prominent characteristics of pectins is their ability to form gels. All pectins are composed of D-galactopyranosyl uronic acid units linked through alpha 1,4 linkage. They contain methyl esters and acetyl groups to various degrees and show a typical average molecular size of 1 lakh Daltons. The highest concentration of pectins in the cell wall is seen in the middle lamella, with a gradual decrease from the primary cell wall towards the plasma membrane. Pectin is found in all plants but is most concentrated in citrus fruits like oranges, lemons, grapefruits and apples. Pectin obtained from citrus peel is referred to as citrus pectin. The extraction of pectins can lead to large variations in the chemical structure of the final material. Pectins are industrially extracted from citrus peels and apple pomace by hot acidified water. Extraction conditions of pH 1.5 to 3 and temperature of 60 to 100 degrees Celsius for 0.5 to 6 hours are varied to give the pectins of desired gelling capacity and degree of methylation. The separation of viscous material from the swollen and partially disintegrated plant material, however, remains a problem. Grinding and washing with ethanol are used, but this can lead to co-precipitation with intracellular proteins, starches and nucleic acids. A method by which this contamination may be avoided is by wet ball milling at low temperature. Enzymatic degradation of the pectin is avoided by addition of a surfactant like sodium dodecyl sulfate or SDS. Sodium deoxycholate is used to remove pigments and lipids, while a treatment with 90% dimethyl sulfoxide, that's DMSO, is used to remove the bulk of starch. Dear viewers, now let us discuss the classification of pectins. Methyl esterification is commonly seen in native pectins. Overall, the degree of substitution is known as the degree of esterification or DE. On this basis, pectins are broadly classified into two categories, that is, high methoxyl pectins and low methoxyl pectins. If degree of esterification in pectin chain is greater than 50%, it is called a high methoxyl pectin or HM pectin, while if the degree of esterification in pectin chains is less than 50%, it is a low methoxyl pectin or LM pectin. Low methoxyl pectins undergo gelation due to the ionic linkage via calcium bridge. These calcium bridges are formed between two carboxyl groups belonging to two different chains in close contact. The mechanism involves the formation of junction zones. In case of high methoxyl pectins, the cross-linking of polymer chains involves extensive segments from two or more pectin molecules that lead to the formation of different junction zones. High methoxyl pectins are only useful in standard jams above about 60% soluble solids. Regarding the application of pectins, the joint FAO-WHO Committee on Food Additives has recommended pectin as a safe additive with no limit on acceptable daily intake except as dictated by good manufacturing practices. The common applications of pectins include their use in jams, jellies, preserves and conserves. In addition, pectin is used in preparation of baker's jelly, frozen berries, barbecue sauces and beverages. In pharmaceutical industry, it has been shown to be effective in removing lead and mercury from the gastrointestinal tract and respiratory organs. When injected intravenously, pectin shortens the coagulation time of drawn blood, thus being useful in controlling hemorrhage or local bleeding. It favorably influences the cholesterol levels in blood and acts as a natural prophylactic substance against poisoning with toxic cations. Fourth important class of heteroglycans includes mucopolysaccharides. Mucopolysaccharides are the heteroglycan molecules, also called as glucose aminoglycans. These are complex carbohydrates which contain amino sugars and uronic acids and constitute the mucous secretions of animals. They are acidic in nature and may be rich in sulfate ester groups. The important mucopolysaccharides include chondroitin sulfate, which is present in cartilage, bone, heart valves, tendons, and the cornea of the eye. Heparin, which is an anticoagulant present in blood vessels, liver, lungs, and spleen, and hyaluronic acid, which is a viscous lubricating fluid present in the skin, vitreous humor of eyes, synovial fluid of joints, and umbilical cord of mammals. Mucopolysaccharides also constitute the principal component of the cell wall of many bacteria, consisting of alternating units of N-acetyl muramic acid and N-acetyl glucosamine linked to short peptide chains. Some important mucopolysaccharides are hyaluronic acid or hyaluronate, that is a lubricant in the synovial fluid of joints, gives consistency to vitreous humor and contributes to tensile strength and elasticity of cartilage and tendons. Chondroitin sulfates that contribute to tensile strength and elasticity of cartilage, tendons, ligaments, and walls of aorta. Dermatin sulfate that is found in skin, vessels, heart, and lungs. The functions are mainly related to coagulation and prevention of vascular diseases. Keratin sulfate that has a role to play in cornea, cartilage bones, and a variety of other structures like nails and hair. Heparin that is a potent natural anticoagulant produced in the mast cells, causing antithrombin to bind thrombin, inhibiting blood coagulation. Glucosaminoglycans are synthesized in the endoplasmic reticulum and Golgi. They are degraded by lysosomal hydrolases. A deficiency of one of the hydrolases results in mucopolysaccharidosis. These are hereditary disorders in which glucosaminoglycans accumulate in tissues, causing symptoms such as skeletal and extracellular matrix deformities and mental retardation. Examples of these genetic diseases are Hunter and Hurler syndromes. These diseases caused by different enzyme deficits are characterized by physical deformities, mental retardation, and disturbances in the degradation of heparan sulfate and dermatan sulfate. This was a brief information about heteroglycans. I hope you have understood well. Thank you very much.